Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today we're going to be looking at the nucleus. The nucleus contains a genome and is surrounded by a double membrane called the nuclear envelope. This contains nuclear pores to regulate molecular traffic. In prokaryotes, the genome is accessible to the cytoplasm as there is no nuclear envelope to isolate it. Replication, transcription, and translation all happens in the cytoplasm in prokaryotes. But in eukaryotes, replication, transcription, and splicing happens in the nucleus, whereas only translation happens in the cytoplasm. Eukaryotic genomes are very organized into linear models of a double-stranded DNA. The large size of the genome makes it necessary to split the genome into pieces, and each piece is called a chromosome. Now, yeast have 4 different chromosomes, whereas humans have 23 different chromosomes. Now remember that each chromosome is a pair, so humans in total have 46 chromosomes. Chromosomes have a centromere near the middle to ensure that newly replicated chromosomes are sorted properly during division. And each eukaryotic chromosome also has a special structure at both ends called a telomere. These have a large number of repeats of a specific DNA sequence, and with the help of a special DNA polymerase called telomerase, they maintain the ends of the linear chromosomes during DNA replication. And these actually shorten with each cell division, eventually leading to a cellular senescence. And this means when cells stop dividing. This shortening contributes to the aging process. And telomeres actually limit the number of times a cell can divide. And this prevents uncontrolled cell growth that can lead to cancer. And in cancers, there is a usually unlimited proliferation, which means you can keep dividing even if the telomeres have emptied out. Now, each chromosome has thousands of genes, and these genes can be mapped genetically and physically to the chromosome they reside on and to a specific location on that chromosome, which is called the locus. These genes are regulated by specific promoter and enhancer elements of the gene, but they can also be affected by the position on the chromosome. Now, this depends on the chromatin. So the function of chromatin is to package DNA into a unit capable of fitting within the tight space of a nucleus. Now, some regions of the chromosome are folded into densely packed chromatin called heterochromatin. In this, genes are usually inaccessible and turned off because it's so densely packed. But there are other regions that are in more loosely packed chromatin called euchromatin, and this allows genes to be activated. Now let's talk about the nucleolus. The nucleolus is a region within the nucleus, and this is essentially a ribosome factory. There is no membrane separating the nucleolus from the nucleus, but it consists of loops of DNA, RNA polymerases, ribosomal RNA, and the protein components of the ribosome. The nucleolus is a site of ribosomal RNA transcription by RNA polymerase 1. mRNA and tRNA transcription is performed by other polymerases in other areas of the nucleus. Now, the protein components of the ribosome get attached outside of the nucleolus in the nucleus. And this is because those proteins are translated in the cytoplasm, so they have to get transported into the nucleus from the cytoplasm. So the ribosome was transported partially complete into the nucleus where it gets some of the proteins attached to it, and then it goes into the cytoplasm, and here it fully matures and then activates. Now let's talk about the nuclear envelope. The nuclear envelope is composed of 2 lipid bilayer membranes. The inner nuclear membrane, which faces the nuclear interior, and the outer nuclear membrane, which faces the cytoplasm. Now, the endoplasmic reticulum membrane is at points continuous with the outer nuclear membrane. This makes the interior of the endoplasmic reticulum contiguous with the space between the 2 nuclear membranes. Just in case you don't know, contiguous means sharing a common border. Now, the nuclear envelope is punctuated with large nuclear pores that allow passage of material in and out of the nucleus. Molecules smaller than 60 kilodaltons can freely diffuse from the cytoplasm to the nucleus via these nuclear pores. However, larger proteins and molecules cannot unless they contain a sequence of basic amino acids called the nuclear localization sequence. proteins with this are translated onto cytoplasmic ribosomes and imported into the nucleus. Now, ribosomes are the molecular machines responsible for protein synthesis. A ribosome is made out of RNA and proteins, and each ribosome consists of two separate RNA-protein complexes, which is known as the large and small subunits. The large subunit sits on top of the small subunit, with an RNA template sandwiched between the two. In eukaryotes, ribosomes get their orders for protein synthesis from the nucleus, where portions of DNA, which are genes, are transcribed to make mRNAs, which are called messenger RNAs. An mRNA travels to the ribosome, which uses the information it contains to build a protein with a specific amino acid sequence. This process is called translation. Now, prokaryotes lack a nucleus, so their mRNAs are transcribed in the cytoplasm and can be translated by ribosomes immediately. So eukaryotic ribosomes may be either free, which means they can be floating around in the cytoplasm, or bound, meaning that they are attached to the endoplasmic reticulum or outside of the nuclear envelope. That's it for the nucleus. Next time we'll talk about the endoplasmic reticulum and the Golgi.